Changes or modifications to the 50S ribosomal subunit, i.e. the target binding site, antibiotics, will confer resistance to macrolides and sometimes other classes. Erythromycin ribosome methylation gene, which is found on plasmids, or i.e. small genetic elements which are capable of moving from one bacterium to another and integrating into the host. Copies of the gene are transported to other bacteria via plasmids or transposons through pili-like channels. The gene is incorporated into the new bacterial genome. During the process of protein synthesis, this bacterium will transcribe and translate the genetic code of the gene, resulting in the production of a protein enzyme capable of methylating the 50S ribosomal subunit at a specific position. This altered 50S subunit results in decreased binding affinity for macrolides and other antibiotics such as lincosamides and streptogrammin type B. This pattern of resistance is referred to as the MLS phenotype. Because the macrolide antibiotic is unable to bind to the 50S ribosomal subunit, it is unable to inhibit protein synthesis, and thus the bacteria itself is not harmed, continuing to produce polypeptide chains of amino acids. Mechanism of bacterial resistance to macrolide antibiotics is mediated by efflux pumps. These efflux pumps are encoded by the MEFA gene, which is a transposable element. Because they confer resistance to macrolides and not lincosamides or streptogrammin B, they are referred to as the M phenotype. The efflux pumps are energy dependent and for S. pneumoniae result in moderate levels of resistance. These pumps traverse the cell membrane of the bacteria and function to pump out the macrolide antibiotic after it has entered the bacterium. It should be noted that for other bacteria, such as Staphylococcus aureus for example, a different efflux system, which is plasmid-mediated and encoded by the MSRA gene, results in macrolide resistance as well as lincosamide and streptogrammin resistance in some cases. Despite the presence of these efflux pumps, macrolide antibiotics continue to enter the bacteria. However, once inside the cytoplasm of the bacteria, these efflux pumps actively remove the macrolide antibiotics before they have a chance to reach their target, the 50S ribosomal subunit, and bacterial protein synthesis is unaffected.